are used for MAS in your program. And then there's actually 2 other questions if you want to do them one at a time.
So the marker we use for marker assist seedling selection is PCR-based markers. And all of our marker were developed from SNPs, single nucleotide polymorphism. And we use the melting curve, the assay, and not TaqMan assay, because TaqMan assay is not work well for us using the strawberry crude DNA, because we use the tiny leaf disc to do direct DNA extraction. So normally DNA quality is not good. So any TaqMan assay does not work for us. So but the HRM works really well.
Thank you, Dr. Lee. He actually answered all of the questions. I guess you are seeing the chat box. And then just a reminder for Dr. Guan, his information, his contact information is in the chat box if you guys need to send specific questions to him. There is a question about the post-test link. It has not been posted. You will have to hold tight. We will make sure that that gets put into the chat box. Oh, and as I speak, it is in the chat box right now. So if you want, open your chat box and click on the link for the post-test. We have 5 more minutes until the next session starts, if anybody else has any questions out there. For those of you that are receiving CEUs for this session, please make sure that you're doing the post-test as we are going through today's activities. Debbie, I see your question in the chat box about what Florida is doing to address the trade imbalances with Mexico. And that is a question, if you can directly correspond with him. We did put his contact information in the chat box. If you don't see it, just let us know and I will send it directly to you. You have about a 5-minute break if you need to step away from the presentations. I did have another good question about CEUs. Some people are indicating that they may have to step away for a session, and can they still get CEUs for the sessions you've attended? As long as you have completed the post-evaluation for those CEUs, you will get credit for those sessions. Okay, I'm going to start a brief introduction for our next session. Again, if you need credit for session 1, the link is in the chat box for session 1's post-test evaluation. Please make sure you click on it and answer it. So for the next session, we're going to jump into disease and pest issues. We have 3 different presenters for you. We have Dr. DeSager, and he is going to be talking about the overview of chemical and non-chemical nematode management in vegetables and strawberries. We have Dr. Lahari, and she is going to be discussing insect management in organic strawberries. And then the last presenter will be Dr. Perez, and she will be talking about one of our newer, not really newer, but a new strain of Pestiolopus on strawberries. So with that, we are queuing up our first presenter, and we have 1 minute to go.
Hello everybody, my name is Johan de Seger. I'm a nematologist at University of Florida, based at the Gulf Coast Research and Education Center, and I will be talking to you about chemical and non-chemical nematode management in vegetables and strawberries. So a brief outline of my talk. I'll cover a little bit the research we did here at GCREC Research Farm on cover crops. I'll be talking also about some nematicide trials we did in growers field on tomato and cucurbids, and then briefly I'll finish with talk a little bit about our more recent organic strawberry research here at the Gulf Coast, looking at different cultivars and steam treatment of transplant. So first, we did some cover crop trials here in last year, spring, summer 2020. This was right around the time COVID hit. We just hadn't planted before, and we had really nice stands, as you can see, some pictures here. We planted sun hemp, sorghum Sudan grass, cowpea, sunflower, vetch, and natural fallow, which was a wheat fallow. The vetch, unfortunately, didn't grow well, got choked out by the weeds, so I won't be talking much about that. So these were large strips. These were about 350-foot long plots by 25-foot wide. So here's the nematode root infections that we found on the cover crop roots at the end of the trial in summer. You see some of the cover crops, we did find some root galls, like on cowpeas and especially sunflower. We did not see any galls, nematode root galls on sorghum Sudan, sun hemp. We also extracted nematodes from the roots, and you can see we found root knot and root lesion nematodes. We found a lot of root knot nematodes in the sunflower roots. We found very little in the cowpea roots, despite the fact that we saw galls. On the other hand, the sorghum and the sun hemp did have some reasonable numbers of root lesion nematodes in there, which we did not find or hardly found in the cowpeas and sunflower. This slide shows you the nematode soil counts pre-plant and at the end of the trial. So we had some reasonable numbers of root knot before we planted. No significant difference with any of the treatment. We also had some root lesion already in there, some stubby root nematodes, and also some dagger nematodes. So a fairly diverse plant parasitic nematode population to start with. At the end of the trial, root knot pretty much increased in most of the cover crops, especially in the sunflower, as you can see. Also in the fallow, where it decreased was the sun hemp. So sun hemp, poor host to root knot, this is what you would expect. And that was really, we also saw actually this reduction in the cowpea, which is interesting. So it did show some galls, but apparently it didn't allow much reproduction of this species or combination of species of root knot nematodes. We still have to ID the species that we have in this field, but it looks like it's a mixed population. Root lesion was quite different, so we found very low numbers in all the cover crop except for sun hemp. Now, sun hemp is known to be a pretty good host for lesion nematodes. Stubby root was the highest in the Sudan grass and pretty low in all the other cover crops. And then dagger nematodes kind of decreased for all the cover crops, so we didn't find many at the end of the trial. So after the cover crops, we planted a tomato crop as well as a zucchini. So we made those rows across the cover crop strips. So the cover crop strips went north to south. The vegetable beds went east to west. So each row covered each cover crop twice. So we had in total 6 replicates as far as the previous cover crop treatment. The crops did really well. You know, we didn't fumigate nothing, and we had a really good stand there, good yields. This table gives you the nematode infection and the yield for both tomato and zucchini. You can see we did get some decent galling, about 6, which is about 60% root galls, the highest. So the lowest root galls we found in the sun hemp and the southern pea, the cowpeas as well. So again, although the cowpeas did gall up, it didn't look like they allowed much reproduction of these root knot nematodes in our field. We didn't see any significant difference with in terms of yield. We had really good yields, but the sun hemp, at least numerically, came out as the highest yield in this trial. Interestingly, we did see very low gall ratings in the zucchini squash. The yields were also very good, no significant difference. But typically, cucurbids are considered good hosts for root knot nematodes. So we decided to do a little side trial in this field where we planted some different cucurbit types. We had cucumber, squash, zucchini. We had some different cultivars for each, the seed we had available. And interestingly, zucchini and squash seem to be much less of a host for root knot than cucumber is. We've noticed this before in field trials, and although squash and zucchini are often good hosts to root knot, they're not as consistently a good host as cucumber is. So maybe there are more root knot species specific differences with squash and zucchini. It's definitely something we need to look more into, and hopefully we'll do that in the next year. Quickly, a few tomato trials we did in the past where we were looking at nimids, fluent sulfone, and non-fumigant nematicides. It's been around several years now. We did these trials with Sanjay Shukla from Immokalee and Joe Noling from Lake Alfred, now retired. These were really large plot trials. We did a trial in Immokalee where we compared the Picklor 60 standard, so this is chloropicrin plus talone, compared it with the chloropicrin plus Nimitz treatment, and we didn't get a lot of galling there. The water tables, the high water tables in Immokalee usually, don't allow such high nematode infection that we get in this area. So fairly low gall ratings, but the Picklor Nimitz really look, good and better than the Picklor 60 in this trial. We did another trial nearer to our center in Parrish, deep sand soils where you typically get more root knot damage. We had the same treatments, and we also had a Nimitz by itself, so a non-fumigant treatment and a control, an untreated control. Here you see the gall ratings at mid-season and end of season, and all the treatments reduced the gall ratings, but again, the PIC plus Nimitz kind of stood out as the best treatment. The Nimitz itself was comparable to the Picklor 60 nematicide standard fumigant. The crops look really good. We didn't see a difference in vigor or yield in these trials. Next, a few cucurbit trials we did in spring 2019. One was a squash double crop on strawberries, the picture on the right, and then a cucumber trial on new beds on the left. Different nematodes. In the cucumber, we had the Javanese, which is typically the nematode, or one of the species you will get in vegetables. The northern root knot nematode is the only root knot nematode we find in strawberries, but it will also affect vegetables. You can see some of the stunted zucchini plants already on the right there. Because of nematode infection, we already saw galls on the roots there. So here, it was basically a rescue treatment. Our applications were done 2 to 3 weeks after planting. And the treatments we applied were, we had vellum, we had vidate, we had magistine, which is biological, and then we had cocktails or mixtures of the different nematicides, but we reduced the rates by half for the mixtures. So this is the nematode soil counts early in the season, March, April, so the soil is still pretty cool, and you see pretty high counts for Hapla in the squash. And we do get a reduction when we apply the vellum and the vellum plus vidate. We didn't see much with the other treatment. But for the cucumber trial, the Javanese root knot nematode, the populations are still pretty low. Part of that is because with this field was also fumigated, but typically this early in the season, Hapla, which prefers the cooler soils, tends to have the much higher populations compared to the Javanese, which is more of a tropical root knot nematode. Late season, it looks just the opposite. This is late May or mid-May, I would say. Much warmer soil. Now the Hapla, the blue bars, are all very low. We still get a little bit of reduction with the vellum vidate mix, but even in the check, the populations are really low. So this is just natural reduction because of the warmth. This nematode is just not so active anymore. Just the opposite with the Javanese, where you get really high populations at this point, and we do get some effect of some of the nematicide. The vellum magistine cocktail actually looks to be the best treatment here. This is the root gall ratings for this trial, and we didn't see any effect of the nematicides at this point on the galls, but what I just want to point out here is the high gall ratings for Javanica, the low gall ratings for Hapla. So Javanica is really the more damaging nematodes for your cucurbits in our area. Then in spring 2020, last year, we did another double crop, zucchini double crop. After strawberry, again, we had the northern root knot nematode. And very similar, you get very high counts pre-plant early in the season here of hapla when the soils are cool. At the end of the season, the population naturally declines, right? But you still see an effect of the nematicide in this case. So we had some different treatments, valium, vidate, magistine. We had a combination. Promax is another biological. And, you know, all the treatments in this case actually look pretty good. This is the gall ratings. We had a really good crop, so there's really no damage of this hapla. And we did see some reduction with valium vidate and the vidate plus magistine in terms of galls. So hapla can cause a lot of damage to those double crops, but as soon as the soil warms up, the crop often recovers. So applying a nematicide as you plant is often enough to save your crop when you're dealing with this type of nematode. Very briefly, since last year, we have a new organic certified research field at the Gulf Coast. This is the second year we're doing organic strawberry trials. I'm just going to show you a little bit of data from last year where we evaluated different cultivars, where we looked at transplant heat treatments. We used Natalia Perez's plant sauna that she's been working with to control certain pathogens that come with the transplants. We know certain nematodes can come with the transplants as well, so we wanted to look at the efficacy of that sauna for nematodes as well. We looked at some organic nematicides. I listed them there. We had very low nematode pressure in this field. This was a new field, you know, it had never been cropped before, as far as I know. We had not a lot of nematodes, so we couldn't say much about the effect of the nematicide. We did see some interesting differences in terms of varieties. So in the nematicide trial, overall, the brilliance yielded significantly better than the beauty. In the steam, the transplant heat treatment, steam treatment trial, we saw the same thing. Again, the brilliance, you know, looked better than the beauty. We had sensation in there as well, which was kind of in between. But interestingly also, although the difference was not significant, we did see, you know, a consistently higher numerical yield in the steamed when we use the steam transplant. So steam treatment may be an option, especially for organic growers. It's one of the few things, you know, they can do. So this may be an option for those growers in the future. And that's all I had. Thanks to my lab, thanks to the Gulf Coast Farm crew and the administration staff for all their support. And feel free to reach out to me. I've listed my email there. We have a website, and I listed some older pictures of my lab we need to update because there's always a big turnover. But thank you for your attention.
All right, keep those questions coming in the chat box. Know that we will have the open mic at the end of session 3. And at that point, we will address these questions directly to the presenters. Dr. Lahari is up next, and she is going to be talking about insect management on organic strawberries.
We are going to talk about all the research that's happening in the library lab to support organic strawberry production. We're going to talk about the key arthropod pests, the natural enemies that we are finding around the strawberry fields, the biorational pesticides, and biological control of pests that are being released effectively, such as predatory mites.
But first, let's talk about the pests that are commonly found in strawberries, starting with the chili thrips. The chili thrips are an invasive species, piercing sucking pests from Southeast Asia, and they feed on strawberry leaves, flowers, and fruits. In this picture, you can see the darkening of the leaflet midrib from chili thrips feeding. Intense infestations can lead to leaf curling, leaf necrosis, and plant death, and highly infested fruits look bronzed and cracked from the thrips feeding. Commonly, the larvae can be found hiding underneath the fruit calyx, along with Western flower thrips, which can also be important pests in strawberries. The two-spotted spider mites are also piercing, sucking pests that are common pests of organic strawberries. In this picture, you can see an adult female with 3 of her eggs that are glued to the web that she has spun. Feeding on the strawberry leaflets causes the appearance of stippling, such as the one shown in the picture on the left. And intense populations of the spider mites will lead to the plant being covered with webbing. Another mite species that can cause potential plant injury are the cyclamen mites. Typically, we find cyclamen mites hitchhiking from the nurseries, and they infest the unfolded young strawberry trifoliates. They start feeding on these leaflets, causing stunting of the young leaflets, or what is also known as the compact leaf mass syndrome. Fruits can also be infested, and you can find several eggs of cyclamen mites underneath the fruit calyx, and these infested fruits will also look bronzed and cracked, just like the thrips infested fruits. In this video, you can see an adult mite hatching out of its egg. This adult mite is tan in color, but may quickly turn light orange in color after it has had a meal. And these eggs are typically glued onto the leaf hair, such as the picture shown on the right. There are several natural enemies that are also commonly found around the strawberry fields, starting with minute pirate bugs, big-eyed bugs that are both equally important in thrip suppression, and other predators such as the multicolored Asian lady beetles, the convergent lady beetle. Here you can see a convergent lady beetle laying a mass of eggs, and lacewing eggs that you can see laid underneath the leaves on single stalks, laid in a bunch. The minute pirate bugs are really tiny. You can see the adult here feeding underneath the strawberry fruit calyx, and it's as small as the strawberry seeds themselves. But they know where to look for their prey because the thrips and the mites like to hide underneath the strawberry calyx, the fruit calyx, or the flower calyx. So they know exactly what they're looking for, as do their nymphs. So we have some very effective natural predators present around the strawberry fields here in Florida. To understand their population dynamics, my PhD student Gagandeep Kaur is conducting field trials, and she's being assisted by my other students, Chastity Perry and Marissa Cassové. And in this study, they are using yellow sticky cards and leaf samples to assess the populations of chili thrips, Western flower thrips, and the commonly found natural enemies that I just mentioned, that is the minute pirate bugs, the big-eyed bugs, and the tiny flies that are beautiful metallic in color, and we commonly call them the long-legged flies. So here is a picture of maps from one single field that was sampled between November 2019 to January 2020. And the top layer of maps shows the chili thrips populations. The middle maps show Western flower thrips populations. And the lowest layer of maps shows the natural enemy populations. And from this map, we were able to conclude that the natural enemies were present in and around the fields at all times during November, December, and January, irrespective of the chili thrips populations. And the highest numbers of these natural enemies tended to occur around the field borders. We did see a couple of population peaks in December and then later on in January. But now we know that there are natural enemies present in the ecosystem around the strawberry fields. And so any pesticide use should be decided keeping this fact in mind that there could be some potential ecosystem services already being provided by these natural enemies present around the strawberry fields. A greenhouse study was conducted by my student intern from Earth University, Armand. Armand released chili thrips onto caged strawberry plants in the greenhouse. And then some of the plants were treated with Captiva Prime, some were treated with the predator Amblyseius swerskii, and some were treated with the Radiant. And what we found from the study was that 7 days after treatment, both the Captiva Prime and the predator Amblyseius swerskii were equally effective in suppressing the adult chili thrips populations as the Radiant was. And then the predator actually continued to suppress the larval chili thrips population through 21 days after treatment. And this suppression was as good as the suppression seen in plants that were treated with Radiant. Currently, many strawberry growers in Florida are releasing predatory mites, Phytoceus persimilis, and Amblyseius swerskii, and Neoceus cucumeris. And so some ongoing studies in our lab are also looking at the hand releases or drone releases of these biological control agents to see how we can further improve the field efficacy of these biocontrol organisms. So as a follow-up to the greenhouse study, we are currently conducting a field study in the organic strawberry plots at the research center here at the Gulf Coast. This study is being conducted by my postdoc, Dr. Jonathan O'Hearn. And in this study, Jonathan is testing a number of biorational pesticides. These biorational pesticides include some biopesticides, some biological control agents such as predatory mites, entomopathogenic nematodes, our Captiva Prime, Azira, Mycotrol ESO, PFR 97, so on. So we have about 10 treatments, including a control in this experiment. This experiment was conducted on December 23rd, 2020, right after we collected chili thrips numbers, and we found that there were at least 30 chili thrips at an average on the strawberry plots. Since then, we have been collecting data on chili thrips, adults, and larvae found in the strawberry leaves and flowers for 28 days. And so far, after data analysis, we have concluded that there are good opportunities that can be explored further. For example, the adult and larval chili thrips were effectively controlled by Entrust. And the larval chili thrips were effectively controlled by the predatory mite Neoceus cucumeris. As we all know, December has been a cold month, and so has January, and cucumeris tends to be a better suited predatory mite species compared to swerskii under such cold climate. Another promising biorational pesticide was the entomopathogenic nematode product. Both species of nematodes tested in these trials showed a lot of promise, specifically Steinernema feltii. Other products that showed larval control were Captiva Prime and Azira. Something to note here that these products were applied only once during this field season so far. So we are conducting further investigation to see what the effect would be if we included a second application of each of these products 7 days apart. In addition to these studies, we are also working with the Perez lab, and Dr. Natalia Perez has kindly allowed us to utilize the Thorwald that is able to apply ultraviolet C radiation in open field strawberries. This work is being conducted by my master's student, Joe Montemere. So Joe here can be seen working with Marissa during the daylight. They are attaching eggs of two-spotted spider mites onto live plants. And then they will wait for sunset. And right after sunset, they will wait for another 1 to 2 hours. And then they will treat these beds with 2 doses of ultraviolet C radiation. From preliminary studies done last year, there were 2 different varieties of strawberries, Radiance and Brilliance, that were tested in 2019. And we found some promising results. In this graph, please note that means with the same letters are not significantly different. The y-axis shows two-spotted spider mite egg hatch percentage, and the x-axis shows the dosage that was actually applied in the field at night. That's 200 and 350 joules per square meters, and there were some plots that were left completely untreated. And after observing the percentage hatch, we were able to conclude that 350 joules per meter square effectively suppressed egg hatchability of two-spotted spider mites, so much so that in Brilliance plots, the egg hatch was significantly less compared to the control plots. Control plots had 57 times higher egg hatch, and 26 times higher egg hatch was seen in Radiance plots when we compared the plots treated with 350 joules per meter square ultraviolet C radiation. So this gives us a lot of hope for this technology, which can be used for organic production also. So now we know that there are several natural enemies. In this picture on the left, you can see a hoverfly that actually is a wasp mimic sitting on Biden's flowers, commonly seen around strawberry fields. That's also predatory in nature. These predators are present in and around strawberry fields, and we should be able to utilize the ecosystem services that they provide. And we are also constantly monitoring all the fields for the pests and the products that we can essentially use to keep supporting the production of organic strawberries.
There are several biorational products available in the market that can be used in organic strawberry production. Several species of predatory mites that can be released advantageously in plants or in and there are several natural enemies such as the mining pirate bugs, the big-eyed bugs, the leading that are out and about here to help us and are present at all times around the strawberry farmscape. Thank you so much for listening to me.
All right, we are right on time for the last presenter, Dr. Perez, and she's going to talk about Neopistalotiopus on strawberries, on regular strawberries, pardon me.
Hello, so today I'll provide an update for this season on the new disease caused by Neopistalotiopsis. I would like to start by acknowledging several people in our group who have worked hard collecting all the data I'll be showing today. In particular, our postdoc, Juliana Baggio, our research assistant, Narni Wang, as well as several grad students, Mayara Bolognesi, Marcos Marin, Paulo Melo, Carolina Rebelo, and Adrian Zuniga. By now, I believe most growers are familiar with the symptoms of the disease, which start with the small leaf spots on the leaves. Those spots can quickly expand to blight a significant portion of the leaves under favorable weather conditions. The leaf spots can be easily confused with many other leaf spots out there, so if in doubt, just bring us a sample to the diagnostic clinic for proper identification. The fungus can also cause a fruit rot. In this case, the symptoms are very similar to those caused by anthracnose or Coleotoxicum nacutatum. So again, if not sure, just bring us a sample to the clinic. I would like to spend a minute now going over the timeline on the emergence of this disease. We first had a significant problem in the 2018-19 season. We basically had 5 farms at that time with a significant problem. And the common link among those farms was the nursery source, which was the same nursery in North Carolina. Moving forward to the 1920 season, we had a significant higher number of farms that had a problem. But again, it was easy to see, particularly early in the season, a link with nursery source. In that case, there's 2 nurseries that had an issue, the same nursery in North Carolina, as well as a nursery in Canada that got plant stock from the North Carolina nursery. Moving forward to this season, we basically had the same number of farms that are infected, but the main difference is that we can't really see that link with a nursery source anymore. What we see is that mostly fields that had the problem last season have it again, which unfortunately indicates that it seems the fungus has established in our fields. So what have we learned so far? Well, so one thing we know is that isolates from the recent outbreaks, they are genetically distinct from isolates that we collect historically on strawberry, as well as isolates from strawberry production areas in other regions. I do realize this is small to read, and I don't expect you to. What I basically want to show you, there's 2 clades. The blue one here includes isolates that were collected historically in strawberry fields in Florida and elsewhere. And now the isolates in the green box here are isolates that were collected from recent outbreaks. So based on these genetic differences, we have developed a quick molecular test that we've been using in our diagnostic clinic. So when we receive samples, we can tell whether it's the new and more aggressive species or the new Bacillotropis rosei, which is not as aggressive. We have also confirmed that with pathogenicity tests that those isolates from the recent outbreaks, they're more aggressive than the other species that we had before. So basically in this graph here, we inoculated fruit, and the isolates in the darker orange here, they cause significant more disease than those isolates in the lighter orange here. This graph to the right, it's basically inoculation of leaves. Again, darker blue cause more disease than the lighter blue, which are basically isolates that were collected in the past. It's important to note here also that the symptoms are identical. The difference is that the new isolates cause more symptoms than isolates that we had collected historically. And this probably has to do with what we see in this slide, which basically shows that under the same conditions, the new Bacillotropis species sporulates a lot more than new Bacillotropis rosei. So this is based on 2 different trials, and in both trials, the new species sporulate a lot more. So more spore basically means it can disseminate more to cause more disease. As far as the conditions for growth and sporulation of the fungus, we have shown that the fungus likes more towards warm temperatures. So basically this graph at the top shows the growth of the fungus in a petri dish at different temperatures, and the optimal temperature that we found was around 77, but the fungus does grow well all the way from 50 to 90. As far as the sporulation, how much spores it produces, it does seem to sporulate better at higher temperatures. In this case, 86 was the optimum for sporulation, but again, it does sporulate some all the way from 59 to 86. So putting all together, the fungus likes warm temperatures and it's water spread. So basically those will be the conditions that are favorable for the disease development. And that's pretty much what we have early in the season when we have warm temperatures, and then we turn overhead irrigation for about 10 days for plant establishment. So if the plants come with the pathogen and you have the favorable environment, we know we have a susceptible host, that's when the disease are going to start. You're going to see the first spots and the first symptoms right after plant establishment. And that's basically what we had also as far as conditions in mid to late December in 2019. We basically had a period in December 2019 for 2 weeks, starting around December 14 all the way to the end of December, where we had 9 days where conditions were highly conducive for disease. Those will be those peaks here at the top. And that was just in a period of 2 weeks. And after that period, we basically seeing this in the fields, a lot of leaf spotting and blighting of the leaves. So looking at this season, and we're looking at a much larger period of time here, all the way from November until where we are now. So at the beginning of November, didn't look good. We had the Ito tropical storm that brought quite a bit of rain. We had 3 days of high alerts, highly favorable conditions. And then just about 10 days later, we had another rain event, another couple of days when conditions were favorable. But after that, we got about a month of a break until just before Christmas, where we got a little bit of rain and a couple of days where conditions were favorable, but it was much cooler and it dried much quicker, so it was not as bad. And then we went on another month with cooler, nice weather conditions until about a couple weeks ago, where we had some rain, but again, not very significant because it was much cooler and it dried much quicker. So compared to last season, we basically had 9 days in a period of 2 months where conditions were highly conducive. Compared to last season, we had 9 days in 2 weeks. So basically the weather has been much nicer this season, and that's one of the reasons why we don't see as much disease out there as we had it last year. Here is how the plants look like about 10 days after the tropical storm in our research plots. They all had symptoms, but the brilliance did look a bit more beat up than the sensation plants. And this is basically what we're seeing in our trials. Then brilliance and beauty does seem to be more susceptible than sensation. We have looked at several different cultivars in advanced selections in this trial, including the 1630-128 that we are releasing now as Florida Medallion, as well as the white cultivars or white selections. And those, interestingly, do not seem to be as susceptible as the red ones. The 1678-109 is the one that we're releasing. Moving on now into our fungicide trial. So before we actually started this trial, we screened a lot of materials in vitro and in a petri dish and identified the most effective to include in the field trial. And these are the results for the field trial up to now. The red bar here is our non-treated control, where we inoculated and didn't treat it with anything. The products here in blue were considered not effective, meaning they're not statistically different than the non-treated control. All the treatments here in orange were statistically better than the non-treated control, even though I wouldn't say they're highly effective, because as you can see, most of these only reduce disease to maybe about 50% of what you have in the non-treated control. So among those, we identify the ones that are registered here are Tyram and Switch, which we had already talked about it last season. But we also have Rhyme, Inspire, and Tilt now showing up as options for rotations. Keep in mind that those three are from the same chemical group. They are all FRAC3, so they need to be rotated with a different mode of action. Omega and Bravo were the most effective, but they're not registered for fruit production fields, but could serve as good options for nurseries. Bravo is already registered for nurseries or chlorothalonil, and Omega is on the process of registration for nursery use only. If you want to learn more how we do those trials, check out our YouTube link and the GCRC website. Juliana and I have recorded a video showing how we do these trials. We also looking at heat treatment as a way for nurseries that are getting plant stock from other nurseries that can maybe use a heat treatment to reduce infection that it might be coming with plant stock. So here we looking at a protocol that we have developed, which it's basically 98.6 for an hour, followed by 111 for 4 hours. And what we see in those two graphs for leaf spot and fruit rot is that the heat treatment can significantly reduce the disease, but it does not completely eliminate. But it might be helpful when for nurseries that are getting plant stock from other nurseries that might be suspect. One question we were getting a lot last season was whether these fungus would be able to over summer in Florida. And well, it looks like it can, and it looks like it did, since many of the fields that were affected last season seem to have it again this season, which is unfortunate. So the question now is, where is it surviving in alternative hosts, weeds, soil, crop residue? So we've been taking a lot of samples from all these alternative hosts, weeds, and unfortunately it's a little bit more complex than we anticipated, since Bacillochypsis, it's usually out there in many different crops, but a lot of the samples we're getting from other hosts, they're genetically not the same aggressive strawberry strain, and when we inoculate it on strawberry, they're just not as aggressive. The ones that were finding to be as aggressive, they're usually the ones that were collecting from weeds in the strawberry field during the season, not between seasons. So there's still more to be done here, but one thing we're really looking close now is a crop residue, as I suspect this may be one of the main culprits. I want to finish up with some management recommendations, and the first one, I think most of the growers have already been doing that this season, which is to avoid harvesting and moving equipment through the fields when the plants are wet, because that's how one of the ways you can move pathogen in the fields. I realize this is much easier to do early in the season when you don't have a lot of fruit than once we get in the peak, which is coming soon. Regarding fungicides, the recommendation is that we do rotations among the fungicides that were found to be effective, and particularly during those times when conditions are favorable, those would be your best choices. It's not too early to start thinking and maybe planning strategies for crop inoculum termination and perhaps removal. I know this might be difficult or not feasible to do, but as I just mentioned on my previous slide, I do suspect that a lot of that inoculum is surviving in crop residue that we're incorporating in our fields. And finally, I think we have a better chance to manage these if we start with clean transplant. So we do need to continue to work with our nurseries so the ones that don't have it can stay clean. With that, I would like to thank my team for all their hard work and for keep smiling during many of those Zoom meetings during this pandemic, and also FSREF for the funding to support this work.
Okay, that concludes our session 2 presenters. And if everybody can take a few minutes, go ahead and put your questions in the chat box. I have one so far for Dr. DeSager. And that is, can you apply Nemetz during cropping through the tape? Okay, we'll give Dr. DeSager a few minutes here to chime in. Meanwhile, I'll address some of the other questions. We do have the post-test evaluation. The link is now in the chat box, and also all of everybody's contact for session 2 is in the chat box. Somebody did ask, what is CCA? And that is a certified crop advisor, and they have to get CEUs as well as other industry people. Natalia, I have a question for you, Dr. Perez. How would you characterize the resistance potential of Pesta, and I'm going to screw it up because I'm thinking about Pesta teleopsis, given it carries over from season to season and appears to be more aggressive pathogen? Dr. Perez cannot unmute her mic if.
Yeah, Lisa, this is Johan. I had the same problem when I had a question earlier, so I couldn't unmute, but I'm unmuted now, so maybe I can answer my question.
Okay, so go ahead and then I'll come back to your question, Dr. Perez. So your question was, can you apply Nemetz during the cropping through the tape?
No, it's not on the label. So Nimitz has a pre-plant application restriction on the label. And the reason for that is you can get some phytotoxicity with Nimitz if you apply it during the crop. So it's not on the label. It's not something I would advise you do. There's some other options you can do during the crop. You can apply a product like Vellum, or if it's a vegetable, you could apply Vidate. Or some of the biologicals that I talked about, like Majesty.
Okay, thank you. And Dr. Perez, your question is, how would you characterize the resistance potential of Pestaloiopsis, given it carries over from season to season and appeals to be more aggressive pathogen?
I assume in the resistant potential as in terms of cultivars. Is that the question? I'm not sure, but what I think what I mentioned towards the end is that I think it would be easier for us to control, and not that's easy, but I think it would be a little bit easier if we start with clean plants and get infections from our fields than starting with infected plants plus getting more inoculum from our fields. There are differences in resistance. If the question is more related to cultivars, they do show differences in resistance. We didn't identify any of the current ones. There are totally resistant, but there are definitely some that are more tolerant, I would say. And in particular, some of the wild types that we're looking and some of the genetics there have shown to be completely resistant, but of course that's a long way for the breeding process. So I'm not sure if that answers the question. You're muted, Lisa.
John, did that answer your question?
You can't read the question here. So.
He didn't go into whether it was cultivar driven, more that it stays in the soil from season to season.
Yeah, and that's why I mentioned that I believe maybe we should start looking into crop removal on a way to, instead of incorporating all those plants in the field at the end of the season, if we could maybe remove how they knock on, in particular in the fields that have been severely affected. I have no idea how to do this, but it might be something we need to look at.
Does anybody else have any questions? We have about 7 more minutes for open question session before we take a break for lunch. Could be why we're not hearing much from people. Everybody's contact is in the chat box if you do come up with another question. If you just click on their information and copy it, you can actually paste it to an email and do some follow-up later on. All right, I do have a question. Oops, it just jumped. A question for Dr. Perez. For Pasteuropsis, is more frequently disking in the summer a potential option as well? Unmute yourself.
Sorry. Yeah, that might help, but I'm not sure, because we think that a lot of where those pathogens hide is on the crowns of the strawberry plants. They're very lignified, and even with the disking, it would help if it chopped out those pieces of crowns, but the disking really doesn't. It basically just moves those pieces around. It doesn't really break them up. So I'm not sure if disking more will help. Will help probably the foliage and the vegetation, but not with those pieces of crowns. That's what I think. You're muted, Lisa.
Thank you. Does anybody else have any questions? Please take this opportunity while you have the different researchers available to you firsthand.
And I just want to add that for the growers in the audience, if you have any ideas, I know growers tend to be very creative, very innovative. If you have any ideas you'd like to discuss on how we could maybe destroy or remove that crop residue at the end of the season, I'll be happy to discuss those ideas with you.
Question, I believe, again for you, Dr. Perez. Is there any possibility of Omega getting registered for strawberry nurseries and or fruiting production?
Yes, for strawberry nurseries, no for fruit production. So the process is already moving with IR4, even though it's a slow process. I think we might be maybe a year or two away from having for nursery. That process is moving well. For fruit production, no, I don't think so, because there is an issue with residue of Omega on fruit. And it's registered on blueberries, but it has a 30-day post-harvest interval on blueberries. So that's something that it wouldn't work for our strawberry production system.
Any other questions for Dr. DeSager on chemical controls or non-chemical for nematodes in tomatoes or in vegetables and strawberry, or for Dr. Lahari on insect management of organic vegetables? Great questions out there, everyone. Okay, so that is our morning session, unless you come up with a question real quick. In the afternoon, when we start on, we will be doing vegetable production, and our moderator will be Craig Fry. And at this point, we're going to break. There in the chat box again is the link for the post test. If you can go ahead and fill that out. We will start back up roughly at 12:30 is when we'll open the session, and then we will start our first presentation at 12:45. We will get the pre-test for the afternoon sessions up in the chat box so that you can go ahead and address those questions beforehand. As a reminder, if you are getting CEUs, remember that you must do these post-tests in order to get credit for them. And all of these presentations, morning and afternoon, will be posted on the Gulf Coast YouTube channel, and you will be getting links for those afterwards. Okay, everybody, enjoy your lunch.
Questions in the chat, and we can get them to answer them. All right, to keep things on schedule, we're going to wait until the next session will begin at 2 o'clock. So we have some time for you to take a break. There's also the post test for session number 3, which is the one that the last 3 presentations we heard. You can go ahead and click on that in the chat and do the post test and get your CCA or CEU credits. If there are any questions, please let us know, and we'll go ahead and try to address those. So there's one question for Sam, wondering what hurdles there are to using compact growth tomato hybrids for commercial production.
Yeah, that's a good question. There's a lot of challenges to breeding tomatoes that are smaller in vine stature. As you get to a smaller bush, a smaller plant type, you're obviously restricting somewhat the amount of foliage that you have to support a heavy fruit load. And then also when you're dealing with a concentrated fruit setting pattern, you've got all of your resources going into those fruit all at the same time versus spreading out the demands over a longer window. All right, so I'd say some of the main challenges we're seeing is trying to maintain acceptable yields and maintain desirable fruit sizes. Both of those things are a little bit challenging. We're not taking hits on fruit size that's getting us down to marbles, but there is a small reduction that we see when we start breeding for compact plants. Other things that we might run into in the future would be when you're dealing with plants that are no longer staked and tied, you've got all of that canopy laying right there on the plastic, and there's not as much airflow. So you have the potential for higher disease pressure for some of your diseases, maybe fungal diseases like target spot or bacterial diseases like bacterial spot. Those are things that we're going to have to keep an eye on as we're pushing this forward.
We still have about 15 minutes before the next session begins. Is there any other questions for our two presenters? Please go ahead and write it in the chat. Without any other questions, we'll go ahead and take a break for about 15 minutes until session number 4 begins. Again, if you are hoping to get CCA or CEU credits, you can go ahead and use the link that was posted for session number 3 for the post test, and then we'll be able to give you credits through that.
This month, we're celebrating American.
Hey Gary, I just got an email from one of our attenders saying that the chat is disabled for them. There's a question in the chat wondering if you can use CRISPR to eliminate green shoulder from bacterial wilt resistant inbreds. Is anyone able to answer that for us?
I'll take a stab at that. Can you use CRISPR to eliminate green shoulder? I'd say regardless of whether that's in a bacterial wilt resistant background or not, you should be able to do that. There is, I'm pretty sure, one gene, there may be two genes that have been identified that control green shoulder in tomato. And we know that knocking out that gene, which is what happened naturally through natural mutations, is how we got to the uniform shoulder. So we should be able to use CRISPR to go in and artificially knock out that gene or silence that gene. So yeah, we could do that if we needed to. Looks like another question, it seems to be related to bacterial wilt resistance. Do we know the number of genes at each QTL that's involved with resistance? So a QTL, we didn't get into that as far as what that acronym means. It means quantitative trait locus. So a quantitative trait such as bacterial wilt resistance that's controlled by multiple genes, and then where one of those loci, where one of those locations are, where genes are. No, we do not know how many genes there are at each one of those locations. That's something we would need to study further. There is evidence that there's probably more than one gene at least some of those loci, such as chromosome 6. There may be a cluster of genes there, and we don't know exactly how small or how broad the interval is where those genes are located. That's definitely something to look into in the future. A couple of more questions here related to bacterial wilt resistance is, have we started looking at chemical profiles that may be related to this bacterial wilt resistance? No, that's not something that we've been doing in our lab. It's something that could be done, and there may be other researchers working on that in other places around the world, but we haven't done it. As far as acyl sugars, that's something that's generally produced on the leaf surface. So it's probably unlikely that acyl sugars are involved with bacterial wilt resistance, since this is a soil-borne pathogen in this case. I was also asked about looking at firmness genes in tomato. This would be for the mechanical harvest project. And yes, that is something that we're aiming to do. We've got some really exceptional levels of fruit firmness in some of these materials. And part of Leo's project is to try to get down and find out where those genes or where that gene is located. And we want to be able to use it, not just for the mechanical harvest project, but for other projects as well. So great question there.
Another question is, do you think you'll have to select for resistance specific to various locations in the eastern US?
That's a really good question, Mr. Odell. I know y'all produce up there, up the East Coast into the Virginias. From what we know about the pathogen, we're really dealing with just one type of strain, one type of biovar of bacterial wilt that's in the US. So right now, given the disease pressures we have right now and the nature of the pathogen, we think that the resistance we have would work anywhere in the Southeast. That's a really good question.
We have about 3 or 4 more minutes before our next session begins. Is there any other last questions about genetics and breeding? Here's one question. Have you encountered any problems regarding the growth of the tomato lines after they are transplanted to the field?
Not, so good question. I'm not quite sure if that's intended for me with bacterial wilt or compact growth projects, or if that's intended for Dr. Lee with his CRISPR-driven tomatoes. But I think in any of those respects, we haven't really seen any problems with these materials going to the field.
Yeah, for the hypergator, I think you can buy the service or storage unit even from outside of University of Florida. That's what I understand.
Another question was about resistance in rootstocks. And no, that's not something we've worked with to map, to locate the resistance genes that's in those rootstocks. That's something that'd be very interesting to do. From what I understand of the history of bacterial wilt resistance, pretty much all sources, or at least in commercial type germplasm, are tracing their resistance back to a very small number of materials. And so the chances are the genetics of resistance are fairly common in most of the material that's used commercially. That may not be the case with rootstocks, because oftentimes those go back to some more wild species. So I think it is possible that rootstocks could be a source of novel bacterial wilt resistance. We haven't worked with that though.
All right, thank you all for your questions. We appreciate being able to have some discussion at the end. Sorry if there was an issue with the chat. Hopefully it seems like it's working well now. So our next session is going to have a couple different presentations looking at vegetable pest and disease management. First, we'll have Dr. Boy talking about weeds. Then we'll have Dr. Carrillo talking about vegetable pathology, Dr. Valad talking about pathology, and then Dr. Smith talking about vegetable entomology. Again, throughout the presentations, you can go ahead and submit any questions, and those that can be answered just in the chat will be done so, and others will be saved to the end for discussion time. If you're just joining us, I'm going to repost the pretest for CCA and CEU credits in just as soon as I can find them here. And then there'll be post-test will be put in the chat after this session for you to go ahead and complete that. And if you get a 70% or more, you will get your CCA and CEU credits. They'll also be sent out by email later on today.
To begin with, let's discuss some of the general production methodologies. We started off with by preparing the field, disking it, harrowing it, and leveling it. Then the next step is we use a hiller to pull up a false bed. After that step, we go through with a standard fumigation rig like growers throughout the state would use, and we apply the fumigants with that rig that is equipped with 3 backward swept shanks. And we apply different rates, different combinations with the same piece of equipment. The final step is we press the beds again and we cover the beds with a plastic mulch. In every case, and this is important to note, for all of these trials, we used a TIF film. For the first experiment, I want to share with you the effects of different rates of picklor 60. For this trial, each plot was 75 feet of an individual bed. It was a replicated trial with 4 replicates. The treatments were rates of picklor 60 from 0 to 500 in 50 pounds per acre increments, and that is on a treated acre basis. The first graph that I want to show you looks at the effects of picklor 60 rate on purple nutsedge density in the field. If you look at this, the x-axis, that's the line along the bottom, has picklor 60 rate in pounds per acre from 0 up to 500 pounds. The other axis, the y-axis, has purple nutsedge density in the number per square meter. The black dots on this graph represent density in the spring, and then the experiment was also conducted in the fall, and that is the clear dots. And I want you to notice a couple of things. Number one, we did have a higher nutsedge density in the fall than in the spring, and control rates achieved in the spring were greater than that was achieved in the fall. So for example, in the spring, at about 150 pounds per acre, you have almost 100% control of purple nutsedge, whereas in the fall, at 500 pounds per acre, you significantly reduce the nutsedge population, but it's still not completely controlled. So now let's go to the field and let's look at some of the pictures of what those plots look like. In this first image, you can see the non-treated control. And I want you to notice 2 things. First of all, the number of purple nutsedge shoots that have punctured the plastic mulch. It's a very thick population. It's going to compete with the tomatoes, and it's going to make removing that plastic mulch more difficult. I also want you to notice the tomatoes, and we're going to talk about tomato yield a little later, but you can see even in this image that there's a lot of stunting of the tomatoes. We had a lot of disease issues, and it's a good example of why we would fumigate in Florida. The second image is at 150 pounds. This was done in the fall, so if you remember back to the graph, at 150 pounds we didn't have great control, as is reflected in this image with the high density of purple nutsedge still occurring in the plot. In this image you can see the 250 pound rate, and again still a significant number of nutsedge occurring in the plot. At 300 pounds per acre, which you can see in this image, you start to see some purple nutsedge control, and there's a lot of variability still. So you start to see good control in some plots, not as good control in other plots. In the image you're looking at right now, you can still see some purple nutsedge there, but it's obviously better. And if you look at the images, one thing that should really jump out at you is the differences in the tomatoes. They're bigger, the canopy is more full and thicker, and the plants just look a whole lot better. Next image is at 350 pounds. Still, plants look great. Good, fairly decent nutsedge control in these plots. The last rate is 500 pounds per acre, which is, of course, much higher than you would ever use. It's above the label rate. But you do notice pretty good control. If you remember back to the graph again, we did not get 100% nutsedge control even at 500 pounds per acre, but we've had a steady decrease, and you can, as visible in this plot, pretty good control, and the plants still look very good. The second graph I want to show you looks at the effect of picklor 60 rate on yellow nutsedge tuber control. So for this study, we took bags of tubers and we buried them in the field, and then we fumigated. 2 weeks later we removed the tubers, and then we sprout them to test for viability. So basically what we're looking for is at what rate is the fumigant able to kill a non-sprouted tuber, or a tuber that could have sprouted while it was in the soil and while there may have been some fumigant present, but they were buried as non-sprouted tubers. And if you look at this graph, it's the exact same setup as the last one and very similar trends. The only difference is that the yellow nutsedge tubers in the spring had 100% control at every rate. So the non-fumigated tubers, the sprouting was about 55%, so about half the tubers sprouted. at any rate, from 100 pounds to 500 pounds, we had 0 sprouting. But in the fall, again, sprouting where they weren't treated is about 75% roughly. But you can see at 100, 200, 250, still a fair number of those tubers are sprouting. At 300 pounds and any rate higher, you get complete control of the yellow nutsedge tubers. The third graph I want to show you is set up the same as the previous two, but this one now is looking at common purslane seed germination. And what we do is we bury purslane seeds in bags, in porous bags, in the soil. We fumigate. 2 weeks later, we retrieve those bags. We then germinate those seeds in petri dishes in incubation chambers. And if you look at the dark black dots, that's the one we'll look at first. This is seed germination in the spring, and you'll notice we are at about 60% germination when it was not fumigated, and any fumigation rate killed 100% of the seeds. Unfortunately, in the fall, even in the non-treated, we didn't have great germination. It didn't seem to have an effect, probably because of the poor germination rate. But this data, although not perfect, does indicate that Picklor 60, even at fairly low rates of 100 pounds per acre, is able to control purslane seeds. The last graph I want to show you is very similar to the previous ones, where we have the effects of different rates of Picklor 60, but now we're looking at total tomato yield. And you'll notice the general pattern is very similar between seasons, even though there's some differences. The important message here is tomato yields at no fumigation are very low, and even at low fumigation rates, as low as 150 pounds per acre, you get a significant boost in tomato yield in both seasons. If you look at the nonlinear
Sorry, I'll apologize for the technical difficulties. Hopefully we'll get it back up and running shortly.
germination when it was not fumigated, and any fumigation rate killed 100% of the seeds. Unfortunately, in the fall, even in the non-treated, we didn't have great germination. It didn't seem to have an effect, probably because of the poor germination rate. But this data, although not perfect, does indicate that Picklor 60, even at fairly low rates of 100 pounds per acre, is able to control purslane seeds. The last graph I want to show you is very similar to the previous ones, where we have the effects of different rates of Picklor 60, but now we're looking at total tomato yield. And you'll notice the general pattern is very similar between seasons, even though there's some differences. The important message here is tomato yields at no fumigation are very low, and even at low fumigation rates, as low as 150 pounds per acre, you get a significant boost in tomato yield in both seasons. If you look at the nonlinear regression, you can see that there's a continued increase in tomato yield up to around 300 to 350 pounds per acre, at which point it begins to level off and even decrease in one of the seasons. So it seems like to maximize tomato yield in these particular field trials had to apply somewhere around the range of 300 to 350 pounds per acre, even when applied under TIF plastic mulch. The second trial I want to talk to you about looked at different rates of 1,3-dichloropropene, which which I'm going to call telone for the rest of this presentation, as well as different rates of chloropicrin. So how we set this trial up is the initial fumigation was exactly the same as described previously, but now each treatment either had 0 or 15 gallons per acre of telone, 2, and then within each one of those telone rates, the 0 or the 15 gallons per acre, we had 0, 50, 100, 150, or 200 pounds per acre of chloropicrin. And we had every possible combination, including a non-treated control. The first graph I want to show you has fall 2018, spring 2019, and then fall 2019. And we're looking at the nutsedge density, which is again the number per square meter. In every season, you tended to have more nutsedge where we did not have telone applied in the plots. It was only significant, however, in the spring 2019, where you see the blue bar, which represents no telone. You can see it's significantly higher than the red bars, which are 15 gallons per acre of telone, or 1,3-D. I do want to point out that there are differences between the seasons, and those differences are important. This first graph is fall 2018. This is the only season of the 3 that we saw this pattern, but this is a common pattern that we see in many studies that we have conducted here at GCREC. So it's different chloropicrin rates, and this is the number of nutsedge per square meter. The black dots are no telone, and the clear dots are with the presence of telone. So let's look at the black dots first. And what you see is the 0, so the non-fumigated to 50 pounds per acre, you actually see an increase in nutsedge density. And that's not surprising. We often see low rates of chloropicrin can stimulate sprouting, so your nutsedge can actually be worse if you have chloropicrin on its own at low rates. But as the rate of chloropicrin increases, you start to see this decrease in the number of nutsedge. However, if you mix telone in with it, the clear dots, you don't see that pattern. We consistently see better nutsedge control with a mix of telone and chloropicrin, and you can see this really clear curve of decreasing nutsedge as you increase the rate of chloropicrin with 15 gallons per acre of telone. The next graph I want to show you is exact same as the last one, but now we're looking at the spring of 2019. And as you saw in the previous graph, if you look at the dots that are black, that's no telone, you consistently have much higher nutsedge density without telone. But if you have telone, which is the clear dots, you see not only fewer nutsedge emerging, but a much more rapid decrease in the density. So at around 150 pounds of chloropicrin with 15 gallons per acre, we have almost 100% nutsedge control. And again, the message that we want to get across is that combination. Having both telone and chloropicrin in the mix gives you much better nutsedge control than using either of the products. This first image is the non-treated control, and as you would expect, really high density of purple nutsedge. Plants are still fairly small in these pictures, but you can see that purple nutsedge is going to cause some problems. In this image, we have 50 pounds of chloropicrin, still no telone. You can see just as many nutsedge in this picture, and in one of the 3 years, we had significantly more nutsedge where we had put the chloropicrin on its own at 50 pounds. Here you can see 100 pounds of chloropicrin. Still significant number of nutsedge growing in the bed, but you're going to notice now the plants look bigger, they're much healthier looking at the higher rate of chloropicrin. In this image you can see it's 150 pounds of chloropicrin. Still see there's a significant number of nutsedge even at this high rate, but a chloropicrin on its own just is not able to control the nutsedge. The tomato plants look better, they look healthier, but we still have significant weed pressure. Finally, at 200 pounds of chloropicrin per acre, you can tell that the nutsedge density has started to thin out a little bit. As you can see in the graphs, you get decreased density, but still not complete control. There's still a fair number of nutsedge in this plot, and the tomatoes are still looking fairly good. Now we're going to switch and look at 15 gallons per acre of 1,3-dichloropropene. And you'll notice right off that still did not achieve nutsedge control. So chlorpicrin on its own did not control the nutsedge very well, except at the very highest rates, and still did not control it completely. Talone on its own did not control the nutsedge very well. The next image here is 100 pounds of chlorpicrin and 15 gallons per acre of 1,3-dichloropropene, or Talone 2. And what you should notice right away is you have a significant decrease in nutsedge density compared to all the other plots. Not 100% control, but still much less density. And this is in a field with a really vigorous nutsedge population. So this kind of really demonstrates our key point is that you need the combination of both products in order to achieve significant control of this really problematic weed. So at 200 pounds per acre of chlorpicrin, you can see almost no nutsedge at all in the plot. This is 200 pounds of chlorpicrin plus 15 gallons per acre of Talone. So at the higher rate, we're getting really good control. Again, it's important to know that there's a lot of variability between seasons. We see this consistently with combinations of Talone and chlorpicrin. Still working out, trying to figure out what causes that inconsistency. So what can we conclude from these experiments? Well, first of all, looking at Pichlor 60, right now to maximize your yields and achieve decent control, you're looking at around 300 pounds per acre, up to 350 pounds per acre under TIF. Even at that rate, we were seeing some inconsistency in nutsedge control. When we're looking at the ratio trials, there's a few things we can conclude. First off, we can conclude that neither Talone or chlorpicrin on their own control nutsedge as well as when you combine the both of them together. 15 gallons per acre of Talone seems to be adequate. When it comes to chlorpicrin with Talone, it seems to be somewhere around 100 pounds to 150 pounds seems to be the rate that we want. But even at 50 pounds, combined with Talone, you do get some significant nutsedge control. In every case, you do get a significant yield increase once you fumigate, regardless of what ratio you used or regardless of what product. And we saw that consistently across all trials. I hope you've enjoyed this presentation today, and if you have any questions, do not hesitate to contact me.
Hello, thank you for attending this virtual field day. I want to talk about some of my work with the evaluation of biological and conventional pesticides for fusarium wilt management on tomato and watermelon. My name is Joseph Carrillo, and I'm currently a postdoc in Gary Villad's lab. So I want to introduce the pathogen. We're talking about fusarium wilts in vegetable production. This is caused by fusarium oxysporum. They have special forms, which are also called forma specialis. Traditionally, they're named off which host they infect. Currently, there are over 58 known special forms. The first one I'll introduce to you today is on tomato. This is Fusarium oxysporum forma specialis lycoprocessi. I'll refer to it as FOL. This is some of the damage it causes on tomato. You can see death that it has caused and some classic chlorosis and wilting here. It has 3 races, 1, 2, and 3. These races develop through the cultivation of tomato. Over time, we've developed resistance to this pathogen, and currently, most commercial varieties are going to have race 2 resistance, but the pathogen evolves to overcome that, and now a lot of population we have out there are race 3, which are causing some issues for tomato growing. Watermelon is a Fusarium oxysporum forma specialis nivium. You can see some of the damage it causes here. It causes a lot of wilting, early plant death in watermelon. Nivium comes from the Latin word, it means snow, which you typically see on watermelon crowns during some infections. In certain conditions, you'll see the mycelium kind of coming out of the crown, which looks white, kind of like snow. I'm going to call this fawn from here on out. And the races here, there are 4 races, 0, 1, 2, and 3. Again, they arise as a result of our cultivation because we're building resistance against them. Signs and symptoms on tomato. The infection is going to occur through propagules that are in the soil, and they infect the rootlets. The optimal temperature is 82 Fahrenheit. Generally, this is going to be a pathogen that prefers a little bit warmer temperatures. Once it's in the roots, it's going to invade the xylem tissue and invade the water transport. It can cause stenting, but the most classic symptom you're going to see is chlorosis. This is going to affect typically one side of the plant at first, as shown here. And also it can affect upper parts as seen here, but typically it's going to be on one side first more than the other. What you can look for on leaves is this classic asymmetrical chlorosis, which you get this golden color, which you've seen in the previous pictures too, but you'll see it on one side and not the other. This can happen on single leaves, the whole leaflet here, but it'll be asymmetrical as opposed to like nutrient deficiency symptoms, which will be typically symmetrical. And you'll get some brown internal discoloration in this greenhouse experiment I did. This is the non-inoculated control. You can see no browning here, but this is inoculated with full. You see some browning in the tissue, and then on the cross section you can see some of the browning there. Symptoms are usually going to appear later in the season, and they can cause some early ripening of fruit and plant death. On watermelon, we get a similar infection through the soil into the roots. The optimal is going to be 77 to 81 degrees Fahrenheit, which is a little bit cooler than full. Fawn is going to prepare a little bit cooler temperatures. Also invades the xylem, or the water transport system, and these are some of the symptoms you'll see. Some early wilting, which can lead to plant death. You can also have some stunting without wilting, but this picture here kind of has a nice comparison of a healthy individual compared to early on wilted individual. You'll also get some gray in the foliage, chlorosis, and wilting like I mentioned. And of course, just like with full and any other Fusarium oxysporum, that's a pathogen, we're going to get some brown internal discoloration as they invade the xylem tissue. Symptoms usually going to appear 3 to 4 weeks and can cause early death before fruit set. Diagnosing Fusarium wilts typically is going to be diagnosed in the field through scouting using visual symptoms and signs. In the lab, the plant samples and soils can be isolated on media, which we can purify to get pure cultures, just like these two shown here. Typically on culture media that has a base of dextrose, Fusarium oxysporum is going to exhibit this kind of purple color, which you can identify as Fusarium oxysporum. However, molecular identification using DNA has become more useful to us because we can distinguish pathogen versus non-pathogen. There's actually Fusarium oxysporum out there that exist in the soil that aren't pathogens. But this identifying the DNA, which the pathogens have that the non-pathogens don't, allow us to determine if the sample is a pathogen or not. So current management strategies are going to be cultural management, using resistant cultivars, clean transplants, grafting, which can be done in tomato and watermelon, using a scion of your desired fruit quality. The rootstock will have the resistance to the disease. Soil pH, if you keep it on the high side, 6.5 to 7, that's going to reduce your incidence. The Fusarium molds typically prefer more acidic soil. And crop rotation is difficult because of these resting structures that they have called chlamydospores. They can survive up to 10 years in the soil, which makes crop rotation difficult. Chemical management is done through soil fumigation using nematicides. They are also effective on full and other Fusarium molds. Piclor 60 is a common one used, but there are other fumigants available in the Florida Grower Handbook. Chemical fungicides are used in watermelon that are applied through drench or drip, plus a follow-up foliar application. And I'll be talking about that a little bit later. And also biological control is another management strategy. This is the use of organisms to control other organisms. I'll be talking about this a little bit later and how we're trying to approach that. So first I want to talk about a field trial that we conducted in the fall of 2009 and spring of 2020. The watermelon variety Jade Star was inoculated with fawn. Application using 6 treatments. 5 reps was done in the fall and repeated in the spring. The treatments selected were Proline, which is the industry standard, and this was applied through a drip with a follow-up foliar application. And we were experimenting with a new product, Miravis Prime, which we use some low concentrations and high concentrations, which are listed here. We did some, with a low and high concentration, we did drip at planting and then foliar 7 days after. With drench at planting, we did, or with the high concentration, we did drench at planting and then foliar 7 days after. And we also did a drench at planting and then drip 7 days after. So again, this is an experimental treatment with Miravis Prime to see how effective it can be in controlling this fusarium wilt on watermelon. So here, this figure shows the disease progress over time, which is also known as the area under disease progress curve. The comparisons between the groups indicate that Proline and Miravis Prime performed similarly, and they both significantly reduced disease compared to the control here, which is marked with a different letter. So we didn't see any differences in the application methods, but we did see similar effective control of the pathogen as far as reducing the incidence compared to the control. Unfortunately, in this season, we weren't able to harvest because of the wildlife were eating our watermelons. So. But we did repeat in the spring, and we saw some more separations of these different methods of application. In particular, we saw the drench followed up by the foliar application had a significant reduction of disease compared to all other treatment groups. We also saw Proline performed, again, significantly less than control, but significantly better than the Miravis Prime treatments, which were done similarly at the high and low concentration using a drip followed up by a foliar application, as we did with Proline. While the drench applications with Miravis Prime both were more effective than Proline in the spring. And here's some images from the field trials. You can see the inoculated control. We have some plants that succumb to the disease, and overall a little bit less foliage than compared to the Miravis Prime shown here, and also the Proline, which did really well in this trial as well. So we were able to harvest at 7 weeks. We did a little bit earlier than normally, just to combat the wildlife issue we were having before, so we can actually make some comparisons of what we're recovering. We converted these values to tons per acre based on the plot size. The yield was significantly higher in both Miravis Prime treatments that included an initial drench, although the follow-up treatment was different. In this one, it was a follow-up foliar treatment. This one was a follow-up drip, but they performed better than the other groups, definitely better than the yield we got in the control. So conclusions and outlook. Miravis Prime is comparable to Proline in managing fusarium wilt of watermelon. And the average tons per acre increased in Miravis Prime treatments that were initially drenched compared to the Proline treatments. And Miravis Prime offers different frac groups, which are 7 and 12 compared to Proline, which is only 7. And for the future, we're going to evaluate this product
of TIF over VF mulch, and why?
With the TIF, you do retain the fumigants for a longer period of time, or at least to not necessarily a longer period of time, but at a higher concentration for a longer period of time, which means you do get better knot sedge control if you're talking about weeds, and should get a better overall pest control. And in addition to that, even without taking that into fumigants into consideration, you do get better weed control just because of that mulch for reasons we're not really sure of. So overall, we're recommending that people move to a TIF just because of evidence of better overall pest control when it's used versus some of the lower density products.
All right, so we have about 4 minutes to go ahead and take another restroom break or coffee break, whatever it may be. And we'll get started at 3:15 for our final session of the day. Again, the post test is in the chat for session number 4. Gary, can you also post the pre-test for session number 5, just in case some haven't done that yet? You can go ahead and do that over the next couple minutes before we get started. All right, welcome back once again. Thank you all for joining us for the 5th session of the day. It's been a long day on Zoom for some of you who have been with us since the beginning. We've got just 3 presentations left. And yeah, if you have any questions throughout the presentations, feel free to put them in the chats and the presenters can answer them during the sessions, or those that need further explanation can be answered at the end during some of our discussion time. Again, my name is Craig Fry. I am the Commercial Horticulture Extension Agent based out of Hendry County for Southwest Florida, mostly with vegetable crops. Thanks for letting me join you all today. Our first session will be for Dr. Mary Lusk, looking at where excess nutrients come from in Florida waterways. Then we'll be looking at geomatics for Dr. Ebhel Haram. And then finally, we'll be looking at some hop production information from Dr. Agahara.
Hi, I'm Dr. Mary Lusk, and I'm an assistant professor in the Soil and Water Sciences Department at the Gulf Coast REC. Today I'm talking about sources of nutrients to Florida's water bodies. So nutrient pollution is a serious problem all throughout Florida, all throughout the United States, really everywhere in the world. Anywhere you have water, And humans, we have the chance of nutrient pollution in those water bodies. And we're concerned about nutrient pollution because it's the nutrients, when they're in excess in our water bodies, can cause problems like you see in this picture here, the thick algal blooms. And the algal blooms can lead to a lot of other problems, like health problems for humans, as well as fish kills and death of other aquatic wildlife. So just to make sure we're all on the same page when I start out here, when I speak of nutrient pollution, I'm talking primarily of having excess nitrogen and phosphorus in our water bodies. So nitrogen and phosphorus are both essential nutrients. They're needed by all living organisms on Earth. But when we have too much of them, it's a perfect example of too much of a good thing. And when we have too much of that in our water, that's when we see problems again, like those algal blooms that you saw on the previous slide. Nitrogen is commonly the biggest problem in our coastal waters in Florida when we have algal blooms, and phosphorus is usually the culprit for our freshwater lakes like Lake Okeechobee. So some of the goals of my research here at the Gulf Coast REC are to take a closer look at those nutrients that can cause those water quality problems, and to identify the sources of them and how they're moving in the environment and how humans are affecting those nutrients, and the management strategies that we need to adapt in order to prevent nutrient pollution of our water bodies. So we have a couple of different things that we work on in my lab, but when I work on water quality and nutrient issues, these are usually the big questions that I ask. I always like to give this slide when I'm giving presentations in the local area, in the Tampa Bay area, because this shows what we're dealing with in the Tampa Bay estuary. So the Tampa Bay estuary is impaired by excess nitrogen, and that excess nitrogen in the Tampa Bay estuary can cause problems like death of our seagrass, which is very important for the Tampa Bay estuary ecosystem. It can also lead to problems with algal growth, fish kills, eutrophication, which then leads to loss of our fishing and our tourism industries. So a couple of years ago, the Tampa Bay Estuary Program began studying sources of nitrogen to the Tampa Bay water body. And a couple of the sources that stand out here on this pie chart that I wanted to talk specifically about include, first of all, atmospheric deposition. So just 21% of the nitrogen that goes to Tampa Bay comes from rainfall. It's coming from the atmosphere. And this originates by vehicle emissions and industrial emissions like power plant emissions. All of those emissions have nitrogen in them. So it goes up into the air, and it's up there in the atmosphere, and it comes down with rainfall. Big source of nitrogen to Tampa Bay, 21%. The next thing I want to point out is that bottom part of the top pie chart where it says 63% stormwater runoff. More than half of the nitrogen that goes to Tampa Bay comes from stormwater runoff. And that's water that just flows over the landscape every time it rains, makes its way somehow to maybe a stormwater pond, and then from there it flows onward to a river, and from the river then it flows on to Tampa Bay estuary. Now that stormwater runoff can come from a lot of different sources. If you look down at the bottom of the pie chart, you see that 20% of that in our area is coming from residential sources. So these would be residential urban neighborhoods. Then we have a couple of other sources like undeveloped land and commercial and industrial, some agricultural uses. But I will always focus on that piece of the pie chart that's residential stormwater runoff. 20% of the nitrogen going to Tampa Bay from that category. And that's where we do a lot of our research in my lab. We focus on that residential runoff and things that urban residents can do on their own lawn, on their own property, to reduce that runoff and reduce the amount of nitrogen and phosphorus in that runoff. Here's just a quick little study. I'm going to take a few minutes to summarize a quick little study that we recently published that was done in Florida to look at residential nutrient pollution. This study was done in a waterfront community in Florida. All of the homes sit on the water. And they have grass or turf grass that goes from the backyard all the way to the water's front. And we collected stormwater runoff from lawns in this community all throughout the year. And we wanted to compare nutrients, nitrogen and phosphorus, and that stormwater runoff during the dry season and during the rainy season. So the dry season being October to May and the wet season being June to September. We also wanted to see if there was an effect of the local fertilizer ordinance blackout period. So if you're not familiar with that, many places in Florida have a local fertilizer blackout period. During that time, you cannot apply urban turfgrass fertilizers to your lawn. That blackout period is usually from June to September during the rainy season, under the premise that rainy season rains flush out nutrients from the soil, and that if people are fertilizing, that's just going to be an extra source of nutrients that are just going to be flushed out with the rainy season rains. So we wanted to test if that ordinance is effective. So we have samples of runoff during the dry season when there is no blackout period in effect, and during the rainy season when that blackout period is in effect. Our first research question in this study was to compare nutrient concentrations in runoff from different landscape designs. And on the left side of this chart, you see FFL, that stands for Florida Friendly Landscaping. And on the right side, you see conventional. Those are conventional lawns. So the FFL lawns had Florida Friendly Landscaping principles throughout the lawn. They reduced turf, they used vegetation other than turf grass. The homeowners made a conscious effort to reduce fertilizer and pesticide inputs, and a couple of other Florida-friendly landscaping practices. The conventional lawns were just that. They were just nothing but turf grass, highly fertilized, lots of chemical inputs. So the data here, we're looking at NO3 in the blue, which is nitrate nitrogen, ortho-P, which is phosphorus in red, and the orange bars are NH4 or ammonia nitrogen. No statistically significant differences between the two landscape types, although the conventional lawns did have a little bit higher phosphorus and ammonia nitrogen in their runoff. Our second research question was to evaluate the impact of the fertilizer ordinance blackout period. So in this data that's shown on this slide, we're looking at all lawns in the dry season versus the wet season. So again, remember the dry season, there is no fertilizer blackout enacted. People can apply whatever they want, whenever they want. In the wet season, we do have a blackout in effect. So no turf grass fertilizers can be applied during the wet season. In this case, we do see a statistically significant difference, particularly for the orange bar, which was ammonia nitrogen. We actually saw higher levels of ammonia nitrogen during the wet season and the runoff from those lawns than we did during the dry season. And that was surprising because it's contrary to the logic of the fertilizer ordinances. We were expecting that during the wet season, when people cannot apply fertilizer to their lawn, that we would see less nitrogen in the runoff, but we actually saw more ammonia nitrogen. And we hypothesize that the reason for that is because of legacy pools of nitrogen in the soils in those lawns. So in this case, we're not seeing any benefits of the fertilizer ordinance because during the rainy season, we're still getting lots of nutrients that are stored in the soils being washed out. They're not necessarily coming from fertilizer, they've just been sitting for some time in that soil. Our third research question was to compare the nitrogen isotopic signatures in our samples to known nitrogen sources. So, strange terms here, isotopic signatures. That's just like a fingerprint. It's a tool that we use to identify where nitrogen comes from. We analyze nitrogen in our samples and we look at that isotopic signature or that fingerprint, and we compare it to known culprits or known sources of nitrogen. And in this case, we can compare it to known sources such as atmospheric deposition, again, nitrogen that just comes from rainfall, or maybe NO3 fertilizer, which would be nitrate fertilizers, or NH4, which would be ammonia fertilizers, or maybe soil pools, or maybe even pet waste. That can certainly be a source of nitrogen as well in residential landscapes. So on this chart, all the little squares and circles that you see represent a sample of runoff taken from one of our lawns in our residential neighborhood. And they plot in different places on this graph. Some of them clearly plot within the area of atmospheric deposition. Some of them clearly plot within the area that represents fertilizer. And some of them clearly plot within the area that says they're coming from pet waste. So basically, it just shows you that we have a lot of different sources contributing nitrogen to the runoff in this neighborhood. And I always like to tell the story of this study, how you'll notice there are a good number of points that plot in that pet waste category. We had a homeowner in this study who loved to tell us how they do not apply fertilizer to their lawn. They try really hard to be good environmental stewards of the water bodies around them. So they applied no chemical fertilizers. They do, however, have a beautiful black Labrador retriever that goes poop in the backyard. And guess what? They don't clean up after the beautiful black Labrador retriever. And so when we analyzed runoff from their backyard, where was all the nitrogen coming from? You got it. It was coming from the pet waste. And we were able to show that homeowner this data. And in this case, it was an easy fix, right? It was something that the homeowner could easily take care of just by beginning to pick up the pet waste. So just in summary, the research findings of that quick little study that we just showed you, sources of nitrogen were highly variable in this neighborhood and could not be attributed solely to landscape sources. Remember, just atmospheric deposition was a major contributing source. Pet waste was a large contributor. Soil nutrient pools may be masking benefits of the fertilizer ordinance. So just because we've put a fertilizer ordinance in place doesn't mean we're going to immediately see results in water quality. We need to remember that. And nutrient management needs to account for and consider all sources of nutrients in a landscape. I always make the argument that fertilizer ordinances may be a good first step, but they're not the magic bullet, and we need to realize that there's multiple sources of nutrients moving to our water bodies, and we need management that addresses all sources, not just one. All right, shifting gears just a little bit, I also do a lot of research on stormwater ponds with an eye of identifying ways that we can improve nutrient removal in those ponds in order to prevent nutrient pollution to larger water bodies like streams and estuaries and bays and our coastal waters. So stormwater flows into these ponds, and we assume that when it's in those ponds, something magical and wonderful happens and the nutrients get removed somehow. Unfortunately, there are many stormwater ponds in Florida. They are all in blue on this map. There are 76,000 stormwater ponds in Florida, actually. They represent about 1% of the state's land area. And research shows that at best, average nitrogen removal from stormwater ponds is only about 30 or 40%. Average phosphorus removal is a little bit better at about 70 or 80%, but we're not getting complete removal of either nitrogen or phosphorus in those ponds, and especially not getting very much removal of nitrogen. So one of the things that we're beginning to do some research on in my lab is how to improve the functioning of those ponds. And one of the things that we're really excited about right now is some research that we're starting on vegetation around those ponds to remove nutrients. Intuitively, it makes sense that if we have a buffer zone of aquatic vegetation and wetland plants, sort of in between the water and the turf grass zone, like you can see in this picture here, intuitively it makes sense that the roots of those plants are going to take up some of those nutrients and help clean that water for us. But we really don't have much research on that. And so that's one of the things that we're starting to do some work on. And I'm really excited about that. We're really hoping that this will provide information that will help us to design these stormwater ponds in a smarter way so that we can remove more nitrogen and phosphorus from them. Okay, so that was me very quickly, some of the work that we're doing. We have many other things that we're working on I would love to share with you. If you'd like to talk more, feel free to reach out to me by email. My email address is here, as well as my Twitter. So thanks, everyone. Bye-bye.
Hi, my name is Amr Abdurrahman. I'm Associate Professor of Geomatics at the Gulf Coast Research and Education Center. In this presentation, I will introduce high-resolution image applications in phenotyping and precision agriculture. Before I start introducing different applications where imagery were used, I would like to show a couple of platforms where cameras capable of capturing high-resolution imagery can be mounted on. So in the left picture here, we can see a platform towed by a tractor in the field where cameras are mounted. And also in the right picture, we can see camera mounted on a drone. So those images can be used in different applications such as field scouting, growth monitoring, yield prediction, stress detection, and diagnosis. Now, in the coming few slides, I would like to introduce the main idea behind using high-resolution imagery. And this is how to extract information, color information, and geometric information from these images that can help us to conduct our applications. Okay, let me start explaining how the image is being captured and created. We know that light is not only one piece. We can see on the rainbow that light can be dissolved into multiple colors. So a camera can be designed to capture different parts of this light, different colors. And in this case, we can see response from the plants at different areas of the light or different colors. Not only in the area that we can see with our eyes, but also in areas that we cannot see, such as the near infrared area, which is very, very valuable for plant studies. So moving forward here and to understand what is an image. So basically an image is an array of numbers. So what happens is when the light gets into the lens to the camera, the light goes into an array of pixels that are sensitive to light, and each pixel would react producing a signal. This signal is measured and converted to a number. So each pixel that we have is basically a number, but it is visualized on the monitor as a color or as a grayscale. So this is very interesting because now we can manipulate these colors in order to analyze the images that we are using. And this is basically what we do in image processing. Now let's dig a little bit deeper into our understanding of how to use images in vegetation studies or agricultural studies. Now, each object can reflect differently. And for plants, they reflect very high in the near infrared area of the light and very small in the red area of the light. So the difference between the near infrared and the red response for a plant at a certain pixel in the image can be an indication of how healthy the plant. Healthy plants will give us very high difference, and unhealthy or stressed plants will give us very small difference. So this is one way to study the plants and using the images, and we can differentiate between healthy or non-healthy plants, or even we can differentiate between different types of plants. We saw how to use colors and extract color information from the images. We can also use the images to extract geometrical information, like the size of the objects in the image, the size of the plants. And we use them based on the same principle that we see with our eyes, three-dimensional objects. If we captured images that are overlapped with each other, we can analytically analyze these images, process them in order to reconstruct the geometry of the scene and capture three-dimensional information, and those could be different plant metrics, plant size, like the canopy height, the canopy size. And we can utilize this information also in our agricultural applications. After reconstructing the geometry of the scene using the overlapped images, we can create three-dimensional information and use this information to create one image mosaic of the whole field from individual images. And also we can create one image where each pixel represents the height of the object. And this would be our 3D information from the images to do our applications. Now I will present applications of high resolution imagery. So the first application would be to predict strawberry characteristics that are very useful to help the breeding program, the strawberry breeding program. The second application would be how to use the imagery and information from the imagery in order to model and simulate growth of the plant. And the third application would be to use the information for yield prediction. So the experiment I will be talking about today, it was conducted at the Gulf Coast Research and Education Center, and the experiment is related to phenotyping application. And phenotyping is about extracting important traits that can be used to evaluate the different varieties that are being developed. So this is very essential component in breeding programs such as strawberry breeding. So when we look at the site, so in this small experiment here, we have two beds that has multiple plots, and each one of the plants in each plot represent different variety. And we would like to see if we can use the images to model and predict the information regarding these plants that represent these different varieties. And those can be used by the plant breeders, for example, to evaluate the growth of the plants, the differences between them, canopy sizes, and many other information related to those new varieties that are being developed. So in our experiment, we use cameras, but we also use other information on the ground. So we use surveying equipment and surveying measurements in order to establish ground control points. And those are points that are surveyed by GPS or surveying equipment in order to provide good control, good positioning control, so that when we produce the images, their locations are accurate. And we use the platform towed by a truck, and the images are, and also navigation sensors like GPS is mounted on top of the cameras on the platform. And we use all this information together in order to capture the images and then take the images and pre-process them. So what we captured or what we produced out of these images, well, for each plant, we wanted to capture information like the average height of the plant, the height standard deviation of the plant, canopy volume, canopy area. So we can extract this information from the data processed. After processing this data, after processing the images captured by the platform, and then at the same time, in situ measurements were taken. So each plot is, the plants on each plot is taken out every week, and then this plant is analyzed in order to measure the dry biomass or the leaf area. Those are very important characteristics of the different varieties and different plants, and those are captured by or needed by the breeding program. So we have the image data, and also we have the in situ data, and we have these for different genotypes, and we have them every week on the strawberry season. We developed a GIS, Geographic Information System, or geospatial analysis models in order to extract the information from the plants. What is the size of the plant? But we also needed to delineate the plants, and we cannot do this manually. It's a very, very cumbersome job, so we had to do this automatically. So we developed the algorithms in order to delineate the canopies and also to extract three-dimensional information from each canopy that is delineated. And we did this using traditional geospatial analysis and also using artificial intelligence techniques. So we used artificial intelligence, just providing the image, training the image, and then the computer will produce these delineated canopies automatically. Extracting the three-dimensional information is very interesting too. We developed geospatial analysis models to do that. And what we can see here, we can see three-dimensional views of the canopies. We can fly through the canopies. They are very high resolution. And we can capture, as you can see here in the profiles, the canopies and the height of the canopies. Not only the bits, but also the height of the canopies. So we developed the models to fill in what we are missing. After removing the canopies from the bids, every week we capture the data, fill it out by interpolation, and then subtracting the canopies, the surface of the canopy from the bids as interpolated, and using this information to capture the three-dimensional information of each plant on the bed. This is the results using artificial intelligence to delineate the canopies. So we can see here all these canopies are delineated automatically. So we use the software, we train the models in order to produce this information.
Apologize once again. Please bear with us as we get the audio and video working again.
Now, we also use high resolution images in order for plant simulation. So we thought if we were able to simulate the plants and produce images out of these simulated plants, we can cut on the cycle that we need to go in the field and start capturing images. So we can plan our image capturing to be at a time where it is the most useful. And this can be for disease detection, can be stress evaluation, and all different applications. So we use these high resolution imagery also to observe details of plants and model them, and used software different. So we used the high resolution images of the plants in order to simulate them, to see how the plants are being created and how they are growing. And we use the information captured from the images in order to simulate the growth of a plant. So we can see in one column at the left-hand side, we can see the actual images planted in the field for one variety. And in the right-hand side, we can see the simulated plant for this variety at the same age. And this is very interesting because we can use these simulated plants and we can study the behavior of the plant. We can even create our own synthetic images that reflect the behavior of the plant. So let's take a look at how the simulation works. So this is very much accelerated growth of a plant that is simulated. And as the plant is growing, we can capture snapshots, and those are the snapshots that we are seeing in the figure to the right. All right, so moving forward, we had to compare analytically what we got from the simulation from what is being produced in reality. So we actually measured that, and we saw that a good fit of the parameters that we were able to extract from the images, just the images taken in the field using the tractor, something like the canopy size, for example, and what is being simulated, which is provide evidence that this simulation can reflect some of the reality, and they can be used again to create synthetic information that can help us planning for images, when to capture images, what is the behavior of the plant under different stressors. We also use the same type of information in order to predict yield. So we collected information about the yield and association with the size of the canopy. Not only the size of the canopy, but we had other information, something like the yield from previous week, for example, or previous couple of weeks, the weather conditions. So we used all this information in order to. So we used canopy metrics extracted from high resolution imagery in building strawberry yield prediction models. And we can see in this slide different models with different variables included, including the canopy metrics that we extracted from the images. And we can see that including these canopy metrics, size of the canopies improved the model goodness of fit. Now, what else we can do? We can do many things with these and with this information. We can use these canopy information extracted from the high resolution imagery, spectral or geometric, in order to do many other applications such as disease detection or any other type of stress detection and diagnosis. Thank you so much. I'll be happy to answer your questions.
Hi, my name is Shinsuke Agehara. I'm a plant physiologist at University of Florida, IFAS, Gulf Coast Research and Education Center. Thank you so much for joining the virtual field day. I'm studying how to grow herbs in a subtropical climate. Today, I'm in a herb field at our research center. This is where we are studying different varieties, LED supplemental lighting for flowering control, and different crop management practices. We also monitor disease and pest issues in the herb field. Our climate is very different from the Pacific Northwest, where more than 90% of herbs are produced in the US. But because of the subtropical climate and flowering control using supplemental lighting, we have a unique production system with 2 growing seasons per year. Our spring season runs from February to June, and the fall season is from June to November. I prepared 3 videos for today's field day. The first video will show how herbs grow in spring and fall. The next video will show the harvesting process for herbs. The last video will show how to operate a mobile herb harvester. Finally, I'd like to share some data. Last year, we started a new experiment to test two trellis designs, each at 3 different heights. The variety used in this study was Cascade. Bead trellis produced 23% higher yield than straight trellis in spring. Increasing trellis height from 12 to 18 foot increased yield by 78% in spring and by 49% in fall. 18-foot bead trellis had the highest annual yield, 1,612 pounds per acre, which was about 90% of the average commercial yield in the Pacific Northwest. This year will be the second year for this trial, so we are expecting that the yield can be even higher. In addition, we are currently looking at the economics of each trellis design and conducting additional trials to optimize the crop management. We share a lot of information on Facebook. If you follow us on Facebook, you will get the latest updates on our hops research. Please check out our YouTube channel, IFAS Horticultural Crop Physiology Lab. You can find the videos you watched today, and there are more. If you'd like to get more details on how to grow hops, please check our publications on this link. Thank you so much for watching the videos. Next time I hope to see you here in a hop field.
All right, do we have any questions? If so, please go ahead and put them in the chat. There's one asking about the efficiency of the hop harvester, and looks like Shinsky said it's about 90% of the cones. Shinsky, I have one. Do you know, have you done any economic analysis of hop growing yet? Do we know what the market capacity might be, as well as what our return on per acre might be?
No, we haven't yet, but we are collecting the production cost information, and we are also collecting labor information. And so I think if we have one more year of data, we will have enough data that we can do enterprise budget analysis, so we can find out break-even price and then also break-even yield.
Great. Any other questions? Shinsky, there's one in the chat asking how many farms are currently growing hops in Florida for breweries.
Yeah, that's a good question. That's what we are trying to find out. As far as I know there are at least 7-8 growers, and the biggest hop yard, the biggest commercial hop operation is about 2 acres now. But most of other growers, they are doing between quarter acre to like half acre. But it's expanding. There are a lot of people contacting us about growing hops, and I know there are more than a few growers planning to start growing hops. So I think it's gonna increase in the next couple of years.
Mary, I have a question for you. I was wondering how your research around the Tampa Bay area, do you think that compares with other locations in Florida for what's contributing to the water pollutants?
Yeah, it's a great question. So I have also done a little bit of work on the east coast of Florida, in the Indian River Lagoon area. A lot of the same issues, a lot of the same sources, things are very similar. One thing that's different in Tampa is we have a lot of atmospheric nitrogen. So just, just being the urban area that we are and the way that storms come into the Tampa area, we can get storms in Tampa from across the Gulf of Mexico or across Florida or up from the Caribbean or even down from Alabama. So we have lots of different storm sources, and it just makes it so that we have a lot of atmospheric nitrogen here. So that's a little bit different. But other than that, we're really very similar across the state. We see a lot of the same trends.
Thanks. Another question, I think, for Sinsky is, does the high humidity and temps cause any unexpected pest or disease pressure?
I think that's a very good question, and that was our expectation when we started the project. But surprisingly, we haven't found any major disease damage, and I think it's because hops had never been grown in this area, and the major hop disease is a downy mildew and the powdery mildew, but they are host specific. So we think that we don't have the strains of downy mildew or powdery mildew that infect hops in this area. But that's why we are telling growers, if they want to grow hops in this area, to use tissue culture plants instead of rhizomes, because rhizome can be infected with diseases. So we want to prevent introducing new diseases into the state. So I think if we use tissue culture plants, at least we can minimize the probability of bringing the disease into the state. And for insect, we found some spider mites could be an issue. In the past couple of years, we used some miticide and agrimec and some chemicals. We also use predatory mites, but when we use the chemicals and predatory mites, we are able to control the mites. And I think spider mite is the only problematic pest. We found other insects, but they were all minor.
If there are no other questions, there is a survey that is posted in the chat. This one is to evaluate the whole afternoon session. If you wouldn't mind giving us some feedback, there's just 4 questions in there, so it won't take you long. If you're doing the CCA or CEU credits, you don't need to fill out the survey because that same survey is in the post-test. So again, the post-test for session 5 is also posted here again for you. And then once a reminder, they'll also be sent out in your email if you're not able to do that right now. That should be everything for everything you need. Oh, here's another question about the quality of the hops. Shinsky, what's the feedback you're getting?
Yeah, that's a very important question because quality matters a lot for hops. I think quality, I'm using only one variety for my trial. It's Cascade. It's one of the major commercial varieties grown in the Pacific Northwest. And for Cascade, the important quality parameters are alpha acid, which is the indicator for bitterness, and also essential oil, which is the indicator for aroma. And for alpha acid, we've been getting at least a commercial level, and some season we are getting above commercial level. So alpha acid, the bitterness is good. For essential oil, we found that it depends more on the age, not just for the plant performance. So in the first year, essential oil can be lower than the average, but after second year, we can get the commercial level. So quality is good for Cascade. And Dr. Deng, plant breeder, is testing different varieties, and Cascade is known as dual purpose variety that can provide both bitterness and aroma. And we are testing also bitterness varieties and then aroma varieties. And then they have different quality, quality characteristics, and some varieties are as good as commercial variety, the commercial herbs in the Pacific Northwest.
And the next question is for Mary, wondering if the soil nutrient pools that are being impacted with wet season releases, can it be assumed that those would be depleted eventually given fertilizer bans, or would they be replenished in the dry season when fertilizer is allowed?
Yeah, that's a great question, Mike. I was typing an answer to you. We don't know. That's a great thing. That's probably one of the things we need to look at next. So my short answer to that is we don't really know what's going to happen there. I suspect that the nutrient pools in those soils right now are enough to cause nutrient leaching for a good couple of years, even with fertilizer bans. And if we just start fertilizing again in the dry season, which is the practice right now, as it should be, then those nutrients are just going to be replenished. So the key that I really want to point out, though, is that as long as those soils and those lawns are covered with vegetative cover, this is not really so much of an issue. As long as there's vegetative cover to have growing roots to be taking those nutrients up, we don't really need to be so worried about those nutrient pools in the soil. But we see it everywhere. We see lawns that have bare patches, lawns that are not really lawns, they're more like just some dirt. That's where we really come into trouble, because all the fertilizer in the world is not going to make a bare dirt lawn grow, but also taking away the fertilizer is not going to prevent erosion of that soil every time it rains. And so we're going to see those nutrients just move right into the stormwater flow. So that was a long, convoluted answer, but in answer to your question, most likely we're just going to continue to see those nutrient pools in the soil for a long time.
Any final questions before we end our virtual field day? All right, if not, it looks like Gary keeps posting all the surveys for you. So you can go ahead and do them for us. That'd be helpful to get your feedback, as well as be able to make sure you get the CCA and CU credits if you're interested in getting those. Thank you all for joining us. We appreciate the team behind the scenes that are also working to make this happen smoothly. And thank you all for participating, for joining us today. And hopefully we get to do this in person next year.